Excellent. Hey, everybody. Thank you so much for having me tonight. I'm Stef. I'm currently the founder and CEO of a company called Cradle. We just got started late last year. Before founding Cradle, I was at Google, initially on the social impact team, where I built some of the first elections coverage into Google search. Then on Maps, if you've ever used Google Maps and it tells you something about traffic jams and how busy it is in places, that was a team that I worked on. Fun fact for the Dutch people, actually the first models that would predict traffic jams were trained on the Dutch road network. And so it's the most accurate one that we have in the world. Then went to Android for the car and was able to launch that into the world. And then at Google X, I got to lead and build a team that made chips that do very well at machine learning workloads called TPUs. And then in my last job, I was on the leadership team of Google Research and Machine Intelligence, mostly with a focus on large machine learning models for language processing. In the Netherlands, I did some work, I guess, tangential to WAG. I was one of the founders of Open State Foundation, so we did a lot of work on opening up government data, building APIs on top of public transit. I can still remember the government hackathon. I actually don't remember the year, but we got this like secret data feed from Tram 7 in Amsterdam that we could then put on maps, which was super hilarious. And I studied in Groningen. Cool. I'd love to give a high-level overview of synthetic biology and especially how it's being used to make stuff. Then secondly, I'd love to talk a little bit more about sort of programming biology. And then finally, I'll lift the veil a bit on what we're trying to do at Cradle. Cool. So turns out that biology is quite versatile. It's also quite messy, but increasingly programmable. And I thought it might be good to sort of really quickly run over some of the central concepts. But cells are these small factories. For those of you that like beer or wine, we've been using them for a while to do things like turning sugars into alcohol. But we can also design these cells. And so inside cells are lots of pathways. You can think of them as little factory lines. And these factory lines are made up of enzymes that can perform specific chemical reactions. You code those things by changing the DNA, of which we saw, some demonstration just now through Benchling and the ART project. By changing what kinds of proteins you put into these pathways, as well as changing the DNA of these individual proteins, you can basically make 60% of the stuff that we're consuming today. And what our startup is doing is really helping bioengineers in that process that was already referred to by the previous speaker of what should we be building or designing and putting into these gels and what should we be synthesizing. That market is growing very rapidly, especially since sort of Moderna demonstrated that you can do this quite fast and do it for a lot of people. But it really, at least if you ask me, is likely going to be the next big thing, sort of beyond compute. Health, food and agriculture, and then materials and energy, I think are some really interesting domains. And I brought some examples of where this already is being used at quite a large scale in the world and some of the use cases that really excite me. So the first one is Perfect Day. Perfect Day is a company in the Bay Area. They make dairy or whey protein. And they do this without cows. So they took this filamentous fungi and genetically engineered it to secrete whey protein. Secretion means spitting out that protein from the cell. And what's super cool, and I actually tried this product, it's the same as what you would get out of a cow, but it's animal free. It like tastes, it scoops, it spreads all the same, and it's also lactose free. But maybe, and so it's a good product. It's identical. And then in terms of the impact, obviously significantly easier to produce at larger scale without having such an incredible impact on the environment. So, we use about 30% of our habitable landmass to raise cattle today, which is quite significant. And you can imagine that growing a fungus, which is not very energy dense, and you can just put them in relatively dark places under normal room temperatures, and they will do their business.
Stef, sorry to interrupt. I forgot to ask, do you want questions in between? Can people just raise their hand?
Yeah, sure. That's fine. Yeah, please unmute and just interrupt me.
And is there has there been a number in terms of scalability to compare it with cow production nowadays?
Yeah, definitely. So it's 97% less greenhouse gas emissions compared to a cow, 99% less blue water consumption, and then 60% less energy use as a cow. And then land mass, I mean, is negligible compared to a cow. Plus all of the, animal rights.
For the same amount.
For the same amount, yeah. It's currently still at a price point that is a little bit bigger than, you can't just make raw cow milk, but that's mostly a scale-up problem of there aren't very many large factories yet that make this stuff, and that's really what Perfect Day is trying to do now. You can currently buy it in ice cream, protein shakes, chocolate desserts, and there's some milk in about 5,000 stores in the United States. And because it's just the whey protein, it's like chemically identical as what comes out of the cow, and you don't eat the fungus, right? The fungus is the GMO, and the fungus makes something that's identical to what the cow would make.
And it's patented, I assume.
The bug is patented, but whey protein is natural, so you can't patent that.
No, because I'm thinking we already have a lot of caves in the south of the Netherlands where they grow fungi, and maybe they could use those caves for this as well.
Absolutely, yeah. It depends a little bit on how you use your fungus. So some people will use the fungus itself to make something. Like one of the later examples is from Bolt Threads. They have two products. One that's a fermented product, and the other product is a leather that's based on the actual fungus itself. Gijs asks, no methane? Nope, no methane. Actually, it absorbs CO2 because it's a living organism, and so it's strictly anything that we as living organisms would take as substrate, like as inputs. It's the same for this organism. Sorry, I was saying yes, and then some of it is fermentation, and so fermentation is more like the DSM factory in Delft, where you have large steel tanks where people grow organisms, or Heineken, where you brew beer. So this is an example of quite a mature product or company, and I think this is really awesome, and hopefully we'll do more. But you can do a very similar thing with like fine chemicals, which obviously we associate that with toxicity and not being very nice. But there's a company called Sologen. They make plant nutrients, cleaning materials, and water treatment sanitization products. These products are today made from petrochemicals, so like derivatives from oil. What they do is they genetically engineer a bacterium, and that spits out an enzyme, and then they throw all the enzymes in a reactor, and they do the chemical reactions, so the multi-step chemistry inside that reactor. So still large steel tanks, but sort of a two-step process. But this is a large-scale chemical factory, and what's super cool is that instead of throwing oil or some derivative product of oil in it, the feedstock for this factory is water, sugar, and air, and there's almost a one-to-one conversion. There's no heat. You can do this at room temperature, a little bit above, but like for those who have brewed beer, it's like 24 to 28 C. No toxins and no carcinogens. So that's quite cool. This is actually the first strictly carbon negative chemical plant. So it absorbs carbon from the environment in order to do its work, and the yields are quite high. It's quite mature, so they have a 40,000 liter per day system, and they can deliver globally. You've already seen this one probably, but obviously we're starting to also see a lot of companies that are making medicines by engineering proteins or literally injecting coding mRNA into people. I'm going to skip over this one quickly, but it's, I think, a really awesome example of you can design the spike, right? The little hat that sits on the COVID bacterium virus, and built this little casing around it, get it into the cell, and then have you as the human make your own, in this case, spike protein. But you can imagine you can use this for treating all kinds of other disease as well by having your own body create your medicine. It's quite mature, as we've seen, over 1.4 billion doses have been made in a relatively short amount of time. Bolt Threads, also super cool. So Bolt is in the materials world. They make two products. They make a silk derivative and a leather using a yeast that they genetically modified. And it's super cool because the silk is actually significantly better than what you would normally get out of silk. And so the tensile strength is much higher. It's much more elastic, it's durable, and it's softer. In terms of impact, most clothing that we wear today is from polyester or some other synthetic fiber. And it's not biodegradable. It ends up in large heaps, usually in developing countries. And here, same thing as the previous example, you feed this yeast water, CO2 and sugar, and it will start producing these protein. This is still only in high-end products. So Stella McCartney and a few others have made products with this that you can buy. So in terms of cost competitiveness, it's not there yet, but that's usually mostly a matter of time before we get there. And then John.bio, I think a very different type of example, but John makes a little microbe that you can basically throw into your crop. And instead of feeding it fertilizer, so nitrogen, this will try to fixate nitrogen that's in the air. I think 60% of air already contains a lot of nitrogen. And it can help protect the crop. Nitrogen fertilizers today account for almost 3% of worldwide greenhouse gas emissions. And so this is a super fun thing. Oh, and by the way, it's one of the biggest polluters of our waterways, especially in the Netherlands as well. And then the first prototypes of this are currently being tested. So this is just a little roadshow. I was at the Build with Biology conference. If you have time to make it there next year, it was super awesome. There are like, more boring things like colorants for food and like Turkish Delight that I tried. There was this guy who made this, he put these fluorescent proteins into little bushes, and you could like put them on your driveway. Instead of having lights, you'd have your little bush light up like a glowfish at night and sort of, walk over your walkway. And there's just a lot of really awesome stuff that's being made by people because a lot of the technologies are advancing really quickly in this space. So why is this happening now? And why are we seeing some of these cool examples starting to pop up? So I think the first thing really is demand for bio-based products is going up. And this is mostly due to either you want a product that has better performance, or you're a company and you want to find ways to be more environmental friendly in your product. So to give you another example of a super cool Dutch company, they're called No Palm. They're in Wageningen. They're making palm oil with the microbe without the palm tree. And obviously, palm trees need to grow first. You need to extract the oil from them. They destroy a lot of rainforest. And then you need to move it from near the equator to where we are, so Unilever can make stuff out of it. And so you can imagine that a lot of companies in that space would really love to have better ingredients into their formulations. Second thing, cost of experimentation is really going down. We just talked about the nanopore machine. You can do long sequence reads with like 99% accuracy, and that's only like last year that has become possible. DNA synthesis, you still need to order that today. There's a company called DNA Script from Paris. They actually made like a desktop printer where you can synthesize oligos and sort of do that overnight, which is super cool. And so these sort of cycle times are starting to look even more like what you might find in software development today. Then, and this is really where my team is focusing, computational methods are starting to get better than biologists. So I think the canonical example here is DeepMind at the end of last year published a second version of a machine learning model called AlphaFold. The problem that AlphaFold solves is you have a sequence, and then this thing called a ribosome can sort of fold the whole thing up. It's like a origami master into a protein, and proteins can like cut things apart. They can tie molecules together, and I'll give you some examples of that later. But they were able to predict the 3D structure of these molecules based just on the sequence, where normally you would need to take it into an X-ray that has incredibly high resolution to get a sense of what your protein might look like. And obviously you can imagine for chemists, like organic chemists and protein engineers, it's super useful to know what your thing looks like. It's a little bit like designing a bridge when there wasn't AutoCAD is sort of the days that we were in, and we're starting to get more into a world where you can sort of ahead of time simulate what might happen with your biological that you're designing. And then finally, this market is just exploding in terms of investment. It's partially due to COVID and sort of the rest of the world waking up to the notion that you can engineer biology without everything turning into a freak. Cool. So just to give you a sense of who works in this space. There tend to be a lot of vertical players. So the companies that we just talked about, they make the individual products. DSM is a really great example of a Dutch company that's been doing this forever. Insulin, if you've ever used insulin, it's made by a yeast. And so lots of medicines are made that way. Then there's a bunch of people that are making design platforms. So Benchling is really good at experiment design and management. We're more in the molecular design space, contract researchers. And then at the bottom, you have these companies like Twist that make DNA, or Chera that make protein for you, or a cell line if you're a pharmaceutical. And then obviously all of the machines and stuff you need in the lab. I saw the pipetting heads. Every new hire in our company also needs to go pipette in our lab in Delft. But luckily there's now also machines that are a little bit bigger. And so like 384 well plates, where all of this is done by liquid handlers that are a bit bigger. That's sort of the thing in the middle there. And these are a lot more accessible to companies these days as well. Okay, so let's talk a little bit about how you program a biological system to do something. So let's just first get a little bit of terminology. And who are these humans that make biology happen? They're actually quite advanced people, and they work at sort of different levels of abstraction. They often make up the R&D departments of food, chemical, pharma, like ag types of companies. And what they do is just change DNA all day. And so the protein engineers work at the sort of smallest level of the machine to make changes, which can help things like, hey, I want this molecule to be broken down in a different way, or sort of the individual steps of the factory. Structural biologists that know a lot about, especially if you're making, for example, medicine, where you want to dock to a certain cancer, these people know a lot about like the physical structures of these things. Strain engineers, they're the sort of factory tuners. So they regulate how much of what protein gets made and how fast these things go, making sure that you don't kill the organism in that process. Organic chemists that do a lot of work in understanding the chemistry and the molecular dynamics sort of inside the cell and outside of it. And then there's a whole army of people that do the pipetting and the lab operations, research managers, and then I guess the sort of analysts of biology, we call them bioinformaticians. So what do they normally do? It tends to be a three-step process. So the first thing that you'll see is that a company will make a proof of concept. And this is usually, they found a bug, they made all the changes for it to produce a very tiny amount of the stuff that they want, whether it's, the product is an enzyme or protein that gets secreted, or a small molecule. And then the second step really is to make it sort of economically viable, because you're competing against an industry, if, for example, you're dealing with petrochemicals, that really has optimized the bleep out of itself over the last several decades. And it's very cheap to dig up dinosaur bones and turn them into products today. And so sort of getting to a point where your little cell factory produces enough of the stuff is very long and tedious process. And then it sort of levels off as you get closer to, hey, I can now put it inside a large fermenter, like 40,000 liters, and start growing this stuff. Typically, it costs about $40 million to get to a working product, which is a lot, right? And so, this is, keep that in mind. This is one of the things that we're trying to work on at Cradle. But then as soon as it's in production, it's mostly sort of improving and squeezing the last bits out of it. So what do they do, these people that program these cells? So usually they're trying to change the characteristics of a microbe or an individual enzyme in order to get more out of it. And programming biology is quite hard. And the reason is that most sort of programming languages that we as humans came up with are fully known. They're distinct. And we can use sort of like object-oriented languages, for example, to write them. You can think of DNA as sort of a massive pile of alien programming language that we found out there, but it's only binary. Like it's literally the instruction code, like zeros and ones. But we don't have any higher level abstractions on top of it, like we have with Python. And so the way that they do this is literally by flipping bits. So they go into the DNA and they change and they see what happens, or they look at how has this thing evolved and what kind of changes seem to have been safe in the course of evolution to be made in the past. And then they try and they try and they try. So what are some of the things that they want to do? If you're like a strain engineer, you might want more of a certain protein being expressed. You might want to reduce bottlenecks in your little cell. You might want to reduce oxygen consumption. I love the sort of genetic stability. So if you're a microbe and this bloody human sort of suddenly decided you need to turn sugar into something else and then use 90% of your energy to do that, like evolutionary, that's not a benefit, right? So there's a lot of these little organisms that will just kick out the pathway in a next generation that you're using to make your little thingy. And so you need to find tricks to make it not eject your changes. And at the level of the enzyme, it's sort of make the enzyme go faster, make it more tolerant to heat. You might remember Moderna sort of in the early days were like, yeah, you should sort of store this at -70 C because otherwise it's going to fall apart. They found ways to make it more stable over time, or substrate specificity, which is what kind of things does it eat when it comes in. I'm going to quickly skip over this. So how do they work? They tend to work in cycles that last weeks to months. It depends a little bit on sort of how fast the thing grows and what machines and steps that sit in the middle. If you're in like pharma and you need to test something in a mammalian cell, like cells like we have, they grow much slower than E. coli, for example. And you just have to wait. So they will design candidates. They'll start each round with a set of sequences. They'll put it in a host organism. They then built their DNA. So they built the candidates. They might put it in the gels, as you just saw. They'll assay, so they'll test these candidates on and see if there are certain properties that have improved or not. And this is actually quite hard, I think, also to the previous speaker's point. It's very likely that you're only measuring a proxy of what you want to measure because you can't measure directly inside cells. And then they hope to analyze these results. Yeah. And they go through, let's say, thousands of candidates and hundreds of cycles before they have one of these products into the market. I'm going to skip over this as well, but let's put it together in an example. So let's say you want to sell carotenoids, which is the ingredient inside food that makes things orange. There's effectively 3 ways of doing that. One would be you buy a lot of carrots, you go smush them together, and you go extract the pigments out of them. The other thing you can do is you can chemically synthesize them by putting acetone, butadiene, dialdehyde, and methanol together. At least one of these is a derivative from a petroprocess, and some of them also show up in a lot of cancer research. So that's maybe not the best way of doing it. Today, you can sell synthetic carotenoids for between $2,000 and $7,500 per kilo, depending on sort of how the market is. So let's say you're a biotech company and you would like to make a microbe that can make synthetic carotenoids. So this is what you would do. So you will start with a proof of concept microbe. And first you're going to insert some genes that you found in carrots into a yeast to see if you can make these individual steps that you need to get to beta carotene, which is the stuff that makes stuff orange. And so you start with a source that is present in your microbe, in this case, FPP. And you start to go find all of the little enzymes, which are the squibbly things with the barrels in them and the beta sheets. And the individual enzymes can do chemical transformation. So what you see here on the right-hand side is you start with FPP, that becomes GPP by CRTE. Then GPP by CRTL becomes phytoene. CRTB will turn it into lysopene. CRTH will turn it into beta carotene. And then you can tell the cell to go secrete it, like spit it out, and then you can use gravity to like get it from your fermenter from the bottom. Now, typical problems that you might run into, first is, let's say CRTL goes way too fast. And so there's too much phytene being deposited into the cell. That becomes toxic. The cell dies, doesn't like that. And so you need to make CRTL go slower. That's the thing you could do. Or CRTH might not be temperature stable. And so after your fermentation goes from like 18 C to 20 C and it heats up because a lot of energy is being used, suddenly your entire reaction stalls and nothing happens anymore. That could be because you have a thermally unstable protein. And then finally, you might not want to grow this entire biomass and then filter the beta carotene out. You might want to just have the cell spit it out, in which case you want to find something that's called a chaperone protein. So it's a little protein that tells the cell, this is like garbage, go throw it out. And then there's this little walking protein that will literally shove stuff to the outside of the cell. Cool. So what are we doing? We're on a mission to make it a bit easier to program these things, starting with...
This was a little bit too fast.
Too fast. Okay, I'm going to slow down a bit then.
Because
Let me pause here and say any questions.
Yeah, that would be good. Give also the other people some time to think. No, I'm just wondering, because this is all done in the biosphere, in like a wet lab. I imagine having a pipette, dripping something inside it, and then it will be adopted, hopefully, by the cell. So that sounds really like the old-fashioned bio. Or is the preparation, has that been to, no, I'm thinking about the programming.
Yeah, so the programming part is really the what sequence am I going to put in? So you can either take something that already existed in nature and just dump it in and pray, or you can actually, change the molecule or the enzyme itself to do something nature cannot do, or to give it different properties. So to give you an example, there was a research group a couple of months ago that made an enzyme that can degrade PET, like plastic.
Plastic, yeah.
Yeah. Obviously, plastic doesn't show up in nature. I mean, it does now, but not at evolutionary timescales, right? And so they've completely engineered a new to nature biological that knows how to degrade PET. And in order to do that, you need to come up with novel sequences. And so in this case, that would be I need to bind to these like major groups of molecules that are inside PET, and you can design these things. And so really what you do before you go put it in is thinking about what should you put in. And that's really what I understand under the programming part, the part that you do at your desk.
Okay, that's the AI helping you to figure out what kind of sequencing you need, and what would work best to put in it.
Yeah, or what sequence should I try? So you saw the image of benchling where you saw the C's, the T's, the A's, and the G's, which is like the instructions that you're giving to the cell. It's about figuring out what should be there in order to achieve a certain goal that you have, which could be degrade PET.
Okay, thank you. Anyone else a question about the previous? Because I really want everyone to have this clear. It's so fundamentally changing our world.
It's also, I think, to Roland's point, the biologists tend to, it's like medicine, like they use all kinds of Latin words everywhere. They love naming the parts names. And so, yeah, to your point, it always sounds very complicated, but I hope I was able to make it a bit simpler.
Yeah, but I just wanted to be sure to understand well. Okay, thanks. Just
Cool.
Next
right.
slide.
Let's keep going.
Yeah.
Okay, so we want to make the programming part easier. So benchling has a bunch of tools to make it super easy to, for example, figure out how do you cut it, how do you deliver it into the cell, like make a plasmid. Plasmid is sort of the thing that shoves it into the cell and then that makes the cell accept the DNA. We're a little bit before that, which is what sequence should you be trying? And so we have a fun team, all former Zymergen, which is a large biotech, DSM. If you ever tried stevia, which is a sweetener in drinks like Coke Zero, Harman actually led the team at DSM that made stevia with Saccharomyces, which is effectively baker's yeast. Yellow was one of the first employees at Uber. Then Franzi is a really awesome ML engineer, Daniel as well, and Thunder as well. And then Steve, I just talked about AlphaFold. He was one of the people that started that project at DeepMind. So a lot of what biology has done in the last 10 years is to make the throughput of experimentation higher. So what Roland was showing to us is sort of what you can do in your basement. Then sort of one up as you go to like 96 to 384 well plates. And if you go further up, companies like Amaris, they can do like 1 million experiments every month. And so let's say the number of shots on goal have gotten dramatically better. But if you're a biologist, most of the time your stuff doesn't work. And Roland was already alluding to this as well. The joke one of the founders I interviewed before starting this company made was 98% of my lab experiments don't work. I just don't know which 98%. And it costs about $150 per candidate, like per sequence to try, right? So it's quite an expensive thing, biology, because of all of the manual steps that you just saw and the cooking that happens in the lab, right? What we think is that machine learning could help with specifically increasing the number of designs that work inside of your microbe. And so first of all, that's in avoiding stuff that doesn't work. So you should never try something. Think of it as compilers for biology. Like you should never try something that is that is unstable, or something that won't express well, like where the liposome or the ribosome is like, I don't know what this is, I'm just going to ignore it, or something that cannot be secreted if you want to secrete it, and sort of really predicting that failure ahead of time so people can eliminate those experiments from their batch. And then secondly, we really think that it's possible to find solutions that are quite novel with help from machine learning. So ultimately, because we don't know how to program biology, a lot of it is like searching pieces of DNA that might do what you want. And ML tends to be reasonably good at finding, let's say, novel solutions out there. So to give you a sense, we've sequenced several billions of sequences in organisms, and the only thing we know about it is where it came from and where it was sampled. It's a database called Tremble. Only 0.0000002% of DNA in the world is annotated, so we know something about it, and often we don't know a lot about it. And so there's still this massive library of, let's say, unexplored potential in biology, and we're only scratching the surface right now. So making that library easier to search through is something we think ML can help with. And then finally, a lot of the tooling that biologists use to do this work is all still, let's say, a little bit feels like the stack from the 90s, right? Where it's a lot of stuff that people do locally on their machine. A lot of it is like government-funded postdoc projects where they built different libraries and sort of hacked them together. And so let's say modern web-based software to do this type of design doesn't really exist. And so that's what we're building. Think of us as Figma or Photoshop for people that make proteins, where we help you predict what you should be trying, help them understand why they work in the ways they work. And then because it's on the internet, it's sort of nicely collaborative. We provide you all the tooling that you're already using, and then you can hook it up to, things like benchling, so you can easily try it out in your own lab. Yeah, so that's what we're doing. If you're a biologist or a computer scientist, we'd love for you to join us. If you're actually a molecular biologist, I don't know if they're on the call. If they are on the call, I'm sorry for butchering some parts. I'm a computer scientist, so I'm probably not doing 100% justice to your field. But that said, I think the combination is super fun. And then if you're in a company that's making enzymes or microbes, we'd love to talk to you. Thank you.
Let me unmute myself. Thank you very much. This is amazing, Stef. Before, to give people also the time to come up with some questions, the idea is, because like you said, the libraries are not there yet. People are, of course, very secretive about what they have tried. So what is not working and what is working well. So using the software, does that mean that you also will be collecting this knowledge that you will, by having them use your software, that the results will be fed back to open libraries, so that system can really learn from it?
Yeah, that is the goal. Yeah, so hopefully people, let's take the example of, is something thermally stable or not, right? Will it die under certain temperatures? That's really a global property of the protein, because it's basically a question of where will heat make it break first. That model will get better over time as it gets to observe more examples, and so other people can use that machine learning model, and therefore hopefully in 5 to 10 years from now, there will not be any more unstable protein. That would be the goal.
And have you noticed that the big companies involved now, because there's so much money going on in this area, are they willing to share that knowledge?
Yes, I mean, I think it will be a little bit similar to what we've seen in the past with things like GitHub, and there will definitely be large old pharma companies that will not use this until they really have to. But luckily, there's lots of smaller companies that are basically saying, why would I reinvent the wheel and do all this stuff in-house? So I think it will be a gradual evolution, but there's definitely enough people out there today. I mean, the way that we're currently thinking about it is you could still use it, quote unquote, private, like if you don't want to share it back, but then you also don't get any of the benefits that the community shared back into it. So you get a less performance than everybody else, like something like that. But we're only a few months old, so this is an emerging perspective.
But I already noticed that you got a lot of funding. Congratulations. Because I think it's brilliant. It will work easier.
To Roland's point, it's also very hard. So it's definitely one of those, if it works, it's going to be amazing.
Roland also explained that, like in the Netherlands, we have, someone is entering now. In the Netherlands, we have this legislation against genetically modified organisms. Part of this is done outside cell-free, I read on one of the sheets. Are there big players in the Netherlands?
DSM is a big player. There's a lot of pharma players out there. Maybe to just clarify what the law says. So you cannot use GMOs for human consumption, for example, or as medicine directly. That doesn't mean that the products of genetically modified organisms cannot be used. So for example, if you can demonstrate that your palm oil is identical to palm oil that comes out of a palm tree, then a regulator is not going to stop you to do that. That said, if you're, for example, in the alternative meat space, and you're making a synthetic, like a lab-grown hamburger, there you do definitely get into trouble. My perspective on this is that the law is a bit odd. So there's a method, for example, called error-prone PCR, where you introduce random mutations into an organism that under European law is not considered genetically modified because it's random. When you start thinking about the introductions, the mutations you want to introduce, it's suddenly GMO, and the sort of regulatory environment becomes very tricky. I think their safety is critical for this type of stuff. And so you do definitely need to build safe things. But for example, at the protein level, like proteins don't multiply, proteins don't have, let's say the complexities of whole organisms or viruses or that kind of stuff. And so there's like a world of things that can be won if we engineer biology to help us instead of just destroying the entire outside world in the process.
Super. Anyone else a question? Michiel, can you detect whether a part of the DNA has been replaced? That's a good question. Yeah.
Could you clarify what you mean by that? As in, if you have
You're muted.
Oh, cool. Yes, Michiel, go.
Michiel, you're muted still. Please unmute.
Or the microphone is not hooked up.
No.
Also, feel free to type to clarify your question if...
Yeah, you can see the messages now. Yeah. Ja, Michiel, wat ongezellig. Yeah.
Maybe we can go to the next question, and then Michiel, if you just type a longer version of what you wanted to ask, then we can come back to that.
Well, I can see it from security perspective, that you would let me, so perhaps something has happened to someone that you would like to be able afterwards to check what kind of modifications have been done to, for example, food or to the person itself.
Yeah, for sure. I mean, so that is always detectable in the sense that, I mean, there's a few asterisks in the sense that, sometimes your sequencing is a bit noisy, but there's so much overlap then that you can see where some of the mutations are being made. We have the phylogenetic tree, so like the tree of life, so we have good understanding of how things evolve over time. And obviously in the software that we're developing and in Benchling that Roland was showing us, you have sort of version control for biology, and you get a sense of like what kind of mutations you're introducing and what kind of impact they have. Going back to the safety argument, there's a ton of use cases, but let me just limit it to some of the things that you can do and some of the things that you shouldn't be too scared of. anything at the protein level doesn't multiply, so I wouldn't be too scared of it. It's like any other chemical that you can throw at something, right? The second is if you're at the organism level, or if you're trying something on humans, you should be, or sorry, if you're at the organism level, there's a couple of things that you can do. One is just treat it well and put it in a factory and don't get it out of that factory. Second is most of these organisms actually don't perform very well in nature because you've given them a task that isn't an evolutionary benefit. And so they use most of their energy to do a thing that won't help them multiply. And so evolutionary pressures tend to help there. The third thing you can do is whenever DNA gets synthesized, and this is actually something that happens today, if you order at Twist and let's say Roland would use a viral vector, there's like a little alarm bell that goes off at Twist, and suddenly somebody will go ask Roland, why are you trying to put this weird virus into your art, man? Right. Obviously, there's standards, like our lab in Delft had to be certified. But then the last thing, if you're really, really, really worried about something that if it would get out, gets you in trouble, you can do two things. Either you can build in a kill switch. So you could make it dependent on something you have to feed it that doesn't exist in nature or isn't abundant in nature. And so it just dies if it shows up in nature. Or you do the inverse, which is if it like gets into contact with something that's very abundant in nature, it dies, which would be a second way of doing that. But most people that you talk to that work on biosafety are more worried about the let's say viral angle here, and so like changing existing viruses to make them better and that kind of stuff. And there's at least pattern matching that you can do today. Can you completely avoid it? I think like with chemistry and other things, not 100%, but GMOs have been around in the US for decades now, and the tools to make very nasty things are out there, and what Roland showed you in the deck should be strictly enough to do that.
Okay, question from Dalen, for both of you, also for Roland, any good learning sources, because what you think you both introduced us to is just the tip of the iceberg. What if people want to get deeper into this topic?
I think Roland might have a better answer than I have, because he did the whole hackery stuff.
There's different answers, I think. I think the difficult thing with biology is that it's so much information online that you kind of don't need, so you have to kind of filter through a lot. So if you search for like any enzyme or protein or organism on Wikipedia, you get like 10 A4 pages if you would print it, so it's always too much. So so there is some good resources. In terms of ethical thing, go watch this documentary that I mentioned, and then newer documentaries too, because there's just very good like documentary about the ethics of CRISPR. In terms of biohacking, there's actually a syllabus by the Biohack Academy, which is still online and maintained. You probably can see the previous year still too. So there's an archive. It's like biohackacademy.github.io.
Then they can see my project from 2015.
Yeah, it's probably in there. So I can put it on the chat too, which is good for like when you want to make your own laboratory equipment. But then for biology in general, I'm now just thinking, I'm looking at my bookshelves, maybe. Well, this is one book that everybody reads. It's got like molecular biology of the cell, and it's like this big, but then somehow if you read a couple of chapters.
Don't start there, because you're never going to go back.
No, but like these images are really good. And I mean, the thing is,
maybe you can share the name in the chat as well.
Yeah, you just have to know the basics of biology to not be scared anymore by the words, and then you can go into kind of the details to the level like what Stef is talking about, because otherwise you continue. It's like learning a new language in a way. Yeah,
it is. We just heard to program it. It's like an alien language.
Yes, well, and all the terminology as well, right? Biologists, I think, like medical doctors, have a tendency to name things after Latin stuff, and it makes it very scary.
We are a do-it-yourself community, also focusing on citizen science. Is there anything that those citizen science communities can contribute in this field?
Definitely. I mean, there's a few communities. So the iGEM community, I think, is great. They're doing a lot of good work making sort of parts and let's say reusable components that you could start sharing with each other. You're starting to see the emergence of that will likely make this much more accessible to citizen science, of like liquid handlers like Opentrons that could get you significantly more scale and sort of do much more interesting experiments. I do think I think this space is probably in the same space as we were before the App Store and the Android ecosystem. It is still quite expensive to have a fully fledged wet lab and run experiments there. So I think, some of the work that organizations like VAG are doing, where they make this stuff much more available, I think is super useful. We had this problem as well, like every startup founder you speak to in this space, if you want to build a fully fledged wet lab, it's like almost $1 million if you want to do high throughput, right? And so that is just not accessible to most people. There's a company called ECL that I think is cool, Emerald Cloud Lab. They're trying to do sort of remote controlled laboratories where you can basically run your processes from the cloud, but it's still a bit janky. Those would be some of the resources I would point people to. But it looks like VAG has a pretty well-equipped set of introductory courses.
Yeah, and there was an app in Amsterdam Noord, Holland. We hosted a SenseMakers meetup there, but I think that's not in function anymore. There was this lab with Federico in the no.
Oh, this was pre-pandemic times even. This is a long
a long time ago. So as an answer to Katerina, it is not our main topic, synthetic biology or biohacking, but I think, like Roland says, the VAG is the right place for that. Just approach them and see what the possibilities are also to use the wet lab. Yeah. Any other questions? Otherwise, I don't want to be rude with your time. Then I will have a last question. Simon, sorry.
Yes, my apologies for missing 75% of the presentation. I'll go back to the video with Ernest, but certainly thanks, Manon, for inviting me to this. I've been following the synthetic biology piece for a number of years, albeit very much as an amateur. And so forgive me if you covered this off in the in most of the presentation. But one of the areas that I'm particularly interested in, I find the whole area fascinating, but is in terms of the storing large amounts of immutable data, given the amount of data that's being created, and I work in law enforcement, so actually keeping a lot of high quality records of massive scale. Because I'm conscious, I've seen previous instances of synthetic biology capturing, putting books and audio and all that kind of stuff. The cost of actually transcribing it to the within the synthetic biology ecosystem was quite expensive. But I'm just quite interested to see how that's evolving at the pace of change of that. When you sense that will actually be a usable technology for the purposes of large amounts of data storage.
Stef, is that a question for you?
There's a couple of companies that are trying to work on this. At the time at Google, we were thinking about it too. There's a whole bunch of challenges still. It's definitely not as easy as writing to disk. A company called Twist Bioscience is doing a lot of work in this space. Let me find an article. And they seem to be quite far. In their process. Currently, what I'm mostly seeing is people that are trying to compete with tape, like tape storage.
Yeah, there was a really cool review article about this, I think 1 year ago or 2 years ago, and then like there's now a big spread of technologies and methods for enhanced assignment, and I can find it on my laptop maybe. You can find it probably when you search for like DNA data storage review. And yeah, it's getting cheaper and cheaper too. I mean, it's obviously not there yet, but I can see why they are super interested in using DNA.
Yeah. Super, thank you.
Yeah. Thank you.
Thank you. I think because we're already running late, so I just Google my question. No, I have found it already. I was just curious about the idea of emailing a fax and then printing it out, with which you also enter like the cybersecurity area. You add additional risks to this domain, I think. But looking at the time, I just want to thank you both very much. And we do have a little present, which I cannot hand over personally. Roland, I think we will meet again on one of the live events. But Stef, I'll send it over to Zurich.
You can also ship it to Delft, where we have our lab, or if you haven't bought it yet, please donate to the Ukraine.
No. That's really nice of you. No, we have, let me just get one.
I am curious now.
We have these very cool notebooks that were donated to us. That were donated to us in such a way that we can give our speakers a present.
Cool.
Sodak gave them. So I'll just ship it to you. But
to Delft, yeah, to Delft is good. I'm actually going to be there next week, so that'd be lovely.
Okay. And Delft is on the website?
Yeah, or I'll ping you the address.
Yeah, super. Then thank you both. Enjoy your evening. This was really great to learn in this way. Thanks for sharing your insights. And everyone.